Hey guys, welcome to this video lecture. Today we're going to be talking about the nerve impulse. So prior to today, you got some information about what is a nerve, what is its structure, also what is its internal makeup. And you know that the internal makeup is essential for allowing a nerve to do its job. Nerves, remember, are made up of hundreds and thousands of neurons, which are individual nerve cells. And today we're going to see how does a neuron go from that resting potential that we talked about before to actually firing a signal. So I want you to take just a moment and read and consider the understandings listed here, and then take a moment to think about what do you know about nerve impulses, what questions do you have, and then to define for yourselves resting potential, because understanding resting potential is essential to knowing the rest of this process. Okay, so every time a nerve fires, basically what we're doing is we're going from resting potential to what's called depolarization, where the signal is actually being fired, to repolarization, and back to resting potential. So we've got this cycle going on. This portion here can also be known as the action potential, like I said, the portion of the process where the nerve is actually firing. And for every nerve signal, mineral ions are going to move in and out of the cell to achieve each state that we discussed here: resting potential, depolarization, and repolarization. Okay, so here are the nuts and bolts of each of those processes. Remember that in resting potential, the neuron has an internal negative charge, and so during that time the sodium pumps are closed, sodium channels are closed, and potassium is being constantly pumped out to maintain a negative internal value. But now what's happened is there's been some sort of stimulus at some sort of receptor. So let's say that someone touches your arm. That would be a stimulus that would cause a neuron to fire. When a neuron fires, what happens is the internal chemistry changes drastically. So before it's been maintaining this negative charge, now all of a sudden the amount of potassium flowing out decreases and the sodium channels open up, and sodium flows into the internal portion of the neuron in large amounts, causing all of a sudden the internal chemistry to change and the charge to go up to about positive 40. This state is called being depolarized, or it's the excitation state, and this is the state in which the neuron needs to be in order to send that signal along to the brain where it can be processed, and then you can interpret the meaning of that signal. Okay, so here's kind of a little overview here. The sodium channels open, more sodium channels open, and now we've reached the height of this action potential, or depolarization, and it's followed by a rapid repolarization. And repolarization is a time where the neuron is going to go back to its resting potential. So as we can see, sodium channels are going to close, potassium channels open back up again in order to get back to that negative state, that about negative 70 state. One addition to this information, however, is that following repolarization, the cell actually goes into what we call a refractory period. And so you can see right here that the signal is a little, or the charge is a little bit below negative 70 because the potassium channels are going to be closing and just kind of maintaining a certain state for just a brief period of time. And this repolarization period, this refractory period ensures that nerve signals won't pile up on one another. It's almost like a built-in little break to make sure that there's a little pause before the next signal can come in. The entire repolarization period only takes 0.02 of a second, so this refractory period, like I said, is very, very brief. Okay, so we've seen resting potential, we have seen action potential, which remember, involved depolarization and repolarization. Now, that was just happening at one site on the neuron. In order to get a full-on nerve impulse, the action potential has to be propagated along the entire length of the neuron. So what we have here is we have the axon of a neuron. The signal is going to be moving in this direction. And what has to happen is sodium needs to flow in at every single location. So there's going to be a depolarization and a repolarization at every one of these little locations. And that is going to propagate the signal down the axon. Okay, so local currents, and local meaning at one particular gate, cause each successive part of the axon to reach threshold potential. And threshold potential is just a strong enough signal to actually get this signal moving. So if we look at the center of this diagram, here is where the action potential is currently located. So the previous gate was just open, and now it is in the repolarization stage, getting back to resting potential. This gate is currently in action potential, or depolarized state. And then this one's going to be the next step. So like I said, we've got our action potential moving down the length of the neuron, and this is how it travels. We've got sodium coming in and out and in and out and in and out at every location, moving that signal down the neuron to bring it to the brain eventually, where it will be processed. Though this sounds like a tedious, time-consuming process, it happens incredibly quickly. The impulse can travel about 0.5 meter per millisecond. Okay, and remember, we're less than 2 meters long, so it can travel very, very quickly throughout our body. And there are some features of an axon that are going to make it travel faster. For example, if an axon has a larger diameter, the signal is going to be able to move with greater speed. And then in addition, if an axon is myelinated, which I'm going to talk more about in just a moment, it can travel faster in a myelinated neuron as well. Before I get deeper into myelination, though, I just want to talk about the stimulus. So remember, the repolarization period had that refractory period where the signal goes up, it comes down, and it actually drops just a little bit lower before it goes back to resting potential. So this here is that refractory period where no other signal can pile on. And so we actually use that beneficially almost to limit the amount of stimulus that we can experience. For example, if you receive a weak stimulus, there is an inconsistent incoming of action potentials. So they come irregularly, there's large gaps, whatever else, and so we perceive this as a softer feeling, a weaker feeling. If we have a very strong stimulus, and often this would be pain, the signals are going to be coming in regularly, close together, and it's going to intensify the sensation that we perceive. However, because of this refractory period, there's a limit to how quickly the signals can come in and how quickly they can be experienced. And so this does almost put a little bit of a limit on how we perceive what we're experiencing. Okay, so remember I said the two things: if the axon has a larger diameter, it can travel faster, or if it's myelinated. So some axons have a symbiotic relationship with these cells called Schwann cells. Schwann cells. And Schwann cells, what they do is they wrap themselves around the axon and they insulate it. And they allow the neuron to do what's called a saltatory conduction. And so this is the propagation of action potentials along a myelinated axon, which increases conduction velocity. What myelination does, this is the myelin sheath, is it allows the nerve impulse, instead of coming in at every single gate over and over and over again, it allows the signals to just come in at the breaks between the myelin sheaths, called the nodes of Ranvier. And so instead of the action potential having to come in here, 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 here. It just has to come in here and here. And so it does speed up the velocity of this conduction, as the action potential doesn't have to come in at every gate. One way that we can measure the state of a neuron is using an oscilloscope. An oscilloscope is something that has an electrode that can be inserted into a neuron and measure the state of the internal chemistry of the neuron. So here we get what's called an oscilloscope trace, and this is going to show us the state of polarization. So for example, location A is in resting potential, while this B area, the internal chemistry is positive, so we know that this is the area of the action potential. And so we're able to see what is the internal chemistry by using an oscilloscope. Alright, lastly, here is a diagram of an oscilloscope trace. This is showing an oscilloscope trace for a neuron that has many incoming synapses. So we can see here that there are many different neurons coming in and attaching here. And so we're going to be looking at the effect of all of these. So A and B are both excitatory synapses, so they're going to be bringing signals, while C is an inhibitory, so it's going to be decreasing the effective signals. So here we've got a signal from neuron A, but it's just a small signal, and it's actually not even reaching the threshold, and so it's not causing a full-on action potential to occur. But if we receive signals in quick succession from neuron A, then that would be enough to build an action potential. In addition, if we get signals from neurons A and B together in quick succession, that will also be a possibility to get an action potential from that and sense whatever is causing that sensation. But then we do also have the effect of neuron C, which is an inhibitory neuron, and so what that does is it causes the resting potential to actually be lower than it normally would, and so it takes more activity then to reach threshold and cause an action potential. So if we get signals from A and B and C, we see that we just get this little blip, and again, it's not going to reach that threshold potential. So this is kind of an overview of how neurons fire, how they bring signals to the brain. Super interesting, super intricate and crazy how fast it can go and how effective it is. Please be sure to write down any questions and bring them to class. Thanks.